All right. I feel like with the start of this one, the start of this unit, I always feel like this is about the time where we need to start making like a fifth grade paper chain to count down the number of lectures until the end of the semester. Right? We're about, what is it, 10 out or so, roughly. Maybe we'll put it. Oh, thanks, guys, for those of you that are actually paying attention. All right. So, let me just, there are a couple of things that I want to make sure to address real quick. So for those that are taking the exam today, those that are taking it between 9 and 12, we're going to meet in 312 Sondrager. So it's another one of the labs. It's the one right next to the lounge. So meet me in there. If you're taking it at 3, just come up by a few minutes beforehand. Just come by my office and I'll get you set up. So that's one. 2, tomorrow the plans in lab are to basically repeat what you did on Thursday, hopefully a little bit less chaotic, right? That's always a goal with these experiments. But what I would suggest is that before you get started with the second round, take a look at your results. If you're not able to actually measure what you're intending to measure, you may need to, 1, modify, or 2, rethink the experiment, okay? And if you use tomorrow as a day to just sit and as a group kind of go through and think about how do we make changes to this, that's a productive day in my mind. Okay, so don't feel like you have to have results, you have to be collecting results tomorrow. It can just be analyzing what you have and deciding, do we need to go back to the drawing board and make some changes? Okay, so those are kind of the 2 big things I wanted to mention. And then the last thing to mention is, as for right now, just kind of what are we doing in the last few weeks in here, okay? So in lab, it's fairly straightforward. Independent projects, just to give you a heads up on your radar, there is a progress report due per group, 1 for each group, and I'll work on asking a couple of questions for that progress report. It's basically, where are you at right now? What data have you collected? What observations have you made? It's a 10-point assignment. It's due, I think I set the due date as like 10 o'clock next week on Tuesday. Okay? We don't have lab next week Tuesday. We don't have class next week on Wednesday. So this would be something that you can finish up before you head out for break. Okay, so that's one. And then what are we doing in here? This last section is focused on the immune system, and focused on prepping the immune system, and focused on how do microorganisms avoid immune detection, or how do they cause disease. Now, we're going to spend 3 days talking about immunology and talking about some examples of pathogenicity. So today is just kind of like a broad overview of what we would call the innate immunity. Wednesday is a broad overview of the adaptive immune system. And then Friday, we'll talk about factors or other protein products produced by microorganisms that cause disease outside of the immune system. So we'll talk about toxins, for example. Next week, Monday, I want you guys in your groups, and the goal on Monday next week is to come up with a topic that you're going to lecture to the class about and to come up with an objective for that. Okay, so we'll go over that a little bit more later. I've got stuff already kind of set up with the rubric and everything, but I'll post that so you can take a look at that. And then the first 2 lectures when we get back from break are your lectures. Okay, these are meant to be 4 to 5 minutes apiece with a couple of minutes afterwards for questions. You only have to physically be standing up in front for the questions. Everything else is video. Okay. And they can be on any topic you want to. It does not have to be related to immunology or related to disease. It can be on any topic that we have spent very little time on throughout the semester. Then we'll go into, I've got kind of 2 days planned for epidemiology, but I'll be honest, I never know how I feel about teaching epidemiology. It feels like a bunch of terminology and sometimes doesn't make a lot of sense. So what I would like to do is at the end of this week revisit, do we want to go a couple of more lectures into immunology, or do we want to move on to something else? Okay, so I'm going to allow you guys In this case, this is a true democracy in this one, for you to decide what do we do with those next couple of class periods. And then the last two, vaccination. We'll spend a day talking about the types. We'll spend a day talking about the controversies. And that'll be it for the rest of the year. Okay? We'll talk about final stuff when it approaches. There will be some announcements then. And then I noticed that yesterday, so I would set up the group so that you could enroll yourself into a group, but sometimes people were being enrolled just randomly into a group. If you have an issue, let me know. I can override it, so I can delete you from a group and add you to a new one if need be. Right now, there's a couple of groups of just 2. I ended up just taking one of those groups of 2 and pairing them up. So take a look. I want to get that all kind of figured out by the end of today, if possible, because tomorrow's homework assignment is a group wiki. Okay, so I'd like to try to get that figured out. Any questions? Kind of a broad overview of what we've got left. All right. So 3 main objectives for today. Describe the process of phagocytosis and its contributions to immunity. Describe inflammation and its contributions to immunity. And then how do microorganisms overcome these innate immune responses? And that's where I want to spend kind of the second half of class, is talking about some examples of how microorganisms overcome these systems. But to begin, what I wanted to do is kind of recircle back on the homework a little bit and actually have you guys break into groups of 2 or 3 or 4. And I'm just going to put up a list of terms related to phagocytosis, and I'm going to have you put them in order. And what I want you to do with each of them is either write down or describe in 1 or 2 sentences what's actually happening during those steps. Now, some of you did go this deep with the homework, some did not, and that's okay. I just was looking for complete answers as I described it. Here's where we start to go a little bit more in depth. So when we say, for example, that the first stage of phagocytosis is attachment, what does that actually mean? What is attaching? Okay, so that's where I want to go with it. So here are your terms. Oh. I already gave you this one. Bless you. So, attachment, pathogen killed, phagosome formed, lysosome forms, maturation of phagosome. Those are kind of, and it is less important to memorize these as specific terms for these steps, but more to get an idea of what does this mean, okay? Because even in the slides, the terminology I use is slightly different than this. So take these, put them in order, describe each in 1 to 2 sentences with a couple of other people. We'll go for about 5 minutes here. That has some of this information on it. We can talk about it in detail. All right, so those are the 5 steps. Attachment, the phagosome forms, or this phagocytic vesicle forms. Step 3, matures. Step 4, phagosome fuses with lysosome. Step 5, destruction of the microbe. Okay, so those are our 5 major steps. Let's go ahead and talk about some of the details of these. When attachment's occurring, what is actually attaching? So when we say there's attachment, Ashley? Okay, so a foreign object binding to a receptor on the surface of the macrophage. What is the term that we use to describe those foreign objects? What typically gets recognized by a macrophage? pathogen, and the phrase or the acronym you might see is PAMP. What does that stand for? Yeah, pathogen-associated molecular pattern. And so these are things that are common among microbes, okay? The innate immune system really identifies more kind of like general themes than it recognizes details. That's what the adaptive immune response recognizes as details. So the innate more is just kind of, yeah, it looks like this. It's going to, examples of pathogen-associated molecular patterns might include lipopolysaccharide. It might include teichoic acid on gram-positives. It might include the peptidoglycan. These are things that are not found normally within our human body, and so they are viewed as foreign by the immune system. What does a PAMP bind to? Go ahead. Good. So the PAMPs bind to pattern recognition receptors. There are specific ones known as the toll-like receptors, and those were the first to be identified. There are other types of pattern recognition receptors, but for the purposes of this class, if we just refer to them as PRRs or TLRs, I don't have a problem with either of those. Okay? So first step is that attachment. Once that attachment occurs, what's happening is then the cell begins to form this phagosome, bring that pathogen in. Okay? And really the phagosome is an endocytic vesicle. So it's one where once we have this attachment, the microbe's just being brought into the cell here. After that forms, what's the maturation look like? What is occurring in the phagosome during this maturation step? We might find a decrease in pH, so we might start to see some acidity building up. Okay? And then eventually, and we also start to find other proteins becoming active in that acidic pH as well. But eventually, that phagosome fuses with a lysosome. What does the lysosome have? Or what is the lysosome bringing in here? I don't think that this was particularly explicit in the mini lecture, so this is a good time to cover it. So a couple of things that we find. Lysosomes, one, they have proteases. They have enzymes that break down proteins. And this is nonspecific, meaning that it basically targets any protein. So we might start to see the breakdown of the pathogen or the microbe that's been ingested here. We also find production of reactive oxygen species and nitric oxide, or reactive nitrogen species, sometimes called ROS and RNS. Okay? And we talked about how before, just to kind of bring this back to something we've talked about in the past, organisms that produce catalase are able to break down those reactive oxygen species and protect them. But the amount that's being produced here, the concentration at which those reactive oxygen species and nitric oxide is being produced is oftentimes too much for the cell to handle, in which case the cell further becomes oxidized and dies, gets chopped into small little pieces, and that's the death. We'll talk about what happens with that on Wednesday. Sometimes this material just gets exocytosed from the cell and released into the environment. Sometimes parts of that pathogen are presented. And we'll talk about the importance of that with the adaptive immune response later. Okay, so those are the major steps. Any questions on those? Should we take a look at an example of how a pathogen overcomes one of these? Which one do I want to do? Your choices are Staph aureus, Streptococcus Streptococcus pneumoniae, Listeria, or Mycobacterium? Streptococcus pneumoniae it is. All right. Thanks for your input, guys. All right. So, this goes back to something we've talked about before. Recall Griffith's experiment. We talked about this when we were discussing, gosh, was it... I think we were talking about mutations, but I think we might have even been talking about transformation in particular, horizontal gene transfer. And you'll remember this is an experiment in which maybe the most memorable thing of this is my really crappy mouse drawings. Okay? So Griffith, it takes a mouse. What? Thanks. Takes a mouse, infects a mouse with smooth Streptococcus pneumoniae, they die. Infects with rough Streptococcus pneumoniae, they don't. Okay? And we talked about how the rough were transformed into smooth because they were able to take that DNA from the smooth cells and incorporate it into their own genome. However, there's context behind this. And if we think about this from a medical perspective, what those results, those initial results of Griffith said were that the rough Streptococcus pneumoniae were not able to cause death, but the smooth were. And the question, in other words, smooth were pathogenic, they caused disease, whereas the rough were non-pathogenic. They did not cause disease. And the question becomes, why? So a group of researchers in 2010 took a look at this a little bit further. They were able to look at one of the genes that contributed to this rough phenotype, or that when deleted or when mutated contributed to this rough phenotype. They took those cells, and on the y-axis here are CFU as a percentage of the initial inoculum. And what that means is that they took neutrophils, and they took either smooth or rough Streptococcus pneumoniae, put them into a little petri dish, incubate them together, and then at time 0 and 30 minutes later count how much Streptococcus pneumoniae is still there. How much is still in the media after these 30 minutes of incubation? And so the y-axis then is then a percentage, basically initial versus final, how much was there? Okay, so 100% would mean there's equal amounts. Greater than 100% would mean there's more after the 30 minutes than there were before. Less than that would mean there's fewer after the 30 minutes than there were before. So 2 parts to this. One, I want you to think about what do the results indicate? Or sorry, one, analyze the results, and then part 2, what do the results indicate? Okay, and you may recall what smooth and rough means for Streptococcus pneumoniae. You may not. Okay, if you don't, take a minute to look it up. I'll give you guys about 3 minutes to work on this one, and then we'll talk it out. Okay. Anybody recall what the smooth versus rough, what the actual difference was, Derek?
Smooth had a capsule.
Bingo. Yeah, the smooth have a capsule, and the rough are unable to produce the capsule. And if you recall, one of the things that we've said about capsules is they make organisms resistant to phagocytosis. We'll take a look at how in a minute. But if we look at this data here, what do we notice? What are kind of the general trends? Which population of cells had more at the end of the experiment? Smooth. Why? What might explain that? Dana? Yeah, and the capsule and them not being phagocytized as frequently as those rough cells. Now, Carla said, "Wait, why is there why are these rough remaining at 100%? Shouldn't there be a decrease? And one of the things that we have to keep in mind is this is a dynamic system. That growth, the incubation temperature at which this was run was enough to allow growth of the microorganism at the same time that phagocytosis is occurring. And so what the data here is saying that is, at least in this system, every time there's a binary fission, one of those two cells is being gobbled up. Okay? The results might look much more dramatic if they had 10 times the amount of neutrophils than they do in this particular experiment. But they don't. So what this data says is actually the rough, at a minimum, it says the rough are worse off than the smooth. They don't have a capsule. How might a capsule protect against phagocytosis? Ashley? That's not a bad idea. So you're saying that it prevents the lysosome, those enzymes from breaking down the organism. That's not a bad idea, but if we think of the context of this experiment, they're looking at how many CFU are still in the media. But we'll come back to that one, because that is a mechanism, actually. Abby?
Is it that, it covers the camps?
Ah, exactly. So what happens is the capsule actually kind of, if you remember, these are sugar coats. It's so thick that it blocks those PAMPs from being recognized or being bound by the PRRs. Okay? So in some ways, the capsule is like a cloaking device for the microbe. Yeah. Can we go to your example? Let's take a look at one more. Yeah, so the PAM, so on the, yeah, let me just finish this up then. So the smooth, because they have that capsule, it's as if those PAMPs are being hidden. In the rough, those PAMPs are exposed, and so it allows for the attachment of the neutrophil to the streptococcus at that point. Yeah, thanks for asking that. So here is a slightly different example. I'm going to draw this one out. Mycobacterium. What is characteristic of mycobacterium? Mycolic acid, acid-fast bacteria. All right, keep that in mind. So in this experiment, what they were doing is kind of similar. They were incubating neutrophils with mycobacterium, and I don't know if they were using smegmatis or tuberculosis. It doesn't really matter too much. The properties of the cells are largely the same. And they were looking at, well, how much mycobacterium is there still outside in the media after 4 hours, and how much is there inside of the neutrophils? So they actually took those neutrophils, blew them up, lysed them, and then did a plate count to determine how many mycobacterium are there. And they looked at two populations of mycobacterium. So we'll make, oh, bother. Someday I'll learn. Okay. We'll make this the wild type. And what they observe is there's this decrease. Y-axis is percentage of the original. And we'll make purple color those that cannot make mycolic acid. All right. And so in the case of these, they find both of them have similar dynamics extracellularly. Both of them are gobbled up. Inside, though, is where we notice the differences. So what we find is inside of these cells, wild-type cells survive and persist, whereas those that lack mycolic acid end up increasing in number early on and then decrease. The question becomes why, or what accounts for this difference, I guess, is maybe a better way to ask this. So what's happening inside of a cell, or what's happening inside of a macrophage through the or a neutrophil during the process of phagocytosis? Ashley. And that's the step that really ends up killing the cell, right? And so that's where your attention might be drawn there. So what Ashley said is phagosome fuses with lysosome, and as we've described, that's what actually results in the death of the cell. Okay? And that's where we see the effect here, is actually that these wild-type cells, if we were to look at images of neutrophils infected with mycobacterium, we would find the mycobacterium in those phagolysosomes. What does that say about the mycolic acid? Maybe one way to describe it is the mycolic acid is a virulence factor. It is a protein, or in this case a wax, which contributes to the disease and pathogenicity of the microorganism. How does it do it in this case? What's true about and mycolic acid, what's true about the consistency of it? It's waxy. Has anybody tried pouring water through wax? Does it work? No? Nobody's tried it because they know it won't work, right? What happens if you pour water on wax? The water beads up. Right? And what we find here is that waxy-like mycolic acid is so thick and impenetrable that it actually prevents the diffusion of the reactive oxygen species. It prevents the proteases from even coming into contact with the proteins. And so in this case, just like what we observed with the capsule, that mycolic acid on the outside here is preventing the breakdown of the cell. Not the attachment step, because it appears, at least from data, that mycolic acid is a PAMP, but it's more with what's happening inside of the cell. Okay, so two different mechanisms here. One that involves avoiding attachment, one that involves the breakdown of the microorganism, but both the end result is the same. The amount of microorganism is able to stay high because of its resistance to that process of phagocytosis. Okay. Questions on these two? All right. Kathleen. Yeah. Yeah. No. That one. Actually, not that I recall, but that's a great question. So you'd want like a third one, or probably a third and a fourth bar, which would be smooth and rough in the experiment before and after, but with no neutrophils added. That's a really nice control. Yeah, I don't know what the results would be. I'm sure that it would be similar to this, if not a little bit higher, to say that the capsule is good, but it's not perfect in protecting. That's a really great point. Absolutely. But I don't recall seeing it from the paper. The other results here, we're looking at the complement system. All right. Let's see. Okay, we've got about 10, 11, 12 minutes left. Let's go over inflammation. Major terms with inflammation. Vasodilation. One of the most fun words on the face of the earth to say, diapedesis, because it just sounds like gibberish. Edema and margination. There's not really so much, I mean, there's a little bit of an order to this one, but what I'd rather have you guys do is describe what these events are and what contributes to them. Or what are the role players in these steps? I'll give you about 5 minutes, and we'll do the same thing. We'll cover this after you've spent some time with it. And in part, I think one of the ways to do this is at least just to go back here and talk about the initial stages. So one thing to point out here is that we have wandering macrophages. We also have resident macrophages. And sometimes what's really a pain in the huge rear end about immunology is that anatomy has a say in it, and so some of the naming is really crappy. Okay, so like there are Kupffer cells, which are specialized macrophages, nothing more than that. So the reason why I point this out is because you may sometimes hear the term like alveolar macrophages, which are ones that are located in the lungs, for example. They are resident, and their job is to basically sit below the epidermis or the mucus layer and, and kind of wait for microorganisms to come into the system. So, for example, if you get a cut where you start to bleed as a result of that cut, you've got macrophages there already that are just waiting to basically get a taste of the bacteria or potential pathogens that might be coming in. Okay? And so those resident macrophages are there. And then when we say first responders, we're talking about the first ones that are recruited to this site of infection. Who are the first responders in terms of an infection? It's the neutrophils. And the neutrophils, I think maybe the best way to put it is the neutrophils' main role is phagocytosis. They make up the majority of the white blood cells. They are not involved in antigen presentation. They just gobble up and produce mass amounts of cytokines to intensify this inflammation response. Again, this inflammation is part of the innate immune system because it is general. It occurs any time there is some sort of injury occurring. But also there are many downsides to inflammation as well. So if you know somebody who has psoriasis or rheumatoid arthritis, those are diseases that are in part thought to have a strong connection caused by overactivation of inflammation. Okay? So we actually have ways to silence this process as well. So let's just go through those four terms and talk about the recruitment of neutrophils to a site of infection. So macrophages secrete cytokines. What do these cytokines do? And use the terms that we said. So vasodilation, edema, margination, diabetes. What do cytokines do? Kaylin. Good. They cause the vasodilation in the blood vessels. The way that this occurs is the cytokines actually will bind to the surface of some of these cells on the epithelium, causing them to begin to kind of change shape and to increase the size of the blood vessels. So that's our vasodilation. That's going to increase the flow of red blood cells and white blood cells at that site of inflammation. What else? What other effect does that have on the blood vessels? What's the term edema mean? Derek? Good. It's a buildup of plasma at the wound site. And so another thing that cytokines do here is that they create this spacing, or they create permeability to the blood vessels, allowing for that blood plasma to now move into that site of injury. So this would be the swelling that we oftentimes associate with an injury. Okay. What's the effect of cytokines or chemokines on neutrophils? So neutrophils kind of bounce, roll, and then what happens? Ashley? Okay, so they will eventually squeeze in between the epithelium that lines the vessels into that site of entry. That's the diapedesis. What happens before then? Yeah, they stick to the wall. So the example I've given in the past is like a shark, and as soon as it senses blood, like its mood completely changes. Maybe another way to think of this is if you've ever watched a bloodhound chase after like a scent, and that bloodhound like will just be looking around, and then all of a sudden it picks up a scent and it becomes like super focused on that one thing. That's what's happening here. Is that neutrophil is just bounding along, dancing, and then all of a sudden it's like, holy crap, there's chemokines in the area. And then it starts to move stealthily or slowly towards that area, increasing concentrations of that chemical. In the same way that chemoattractants work for bacterial cells, and it moves towards increasing concentrations of that chemical to the point source of where the infection is occurring. And once there, so that process is the margination, and then the squeezing through, which is the diapedesis. Some books will refer to it as this extravasation. But once it gets there, now the neutrophils start gobbling everything up that they can. PAMPs attached to PRRs, destruction in the phagolysosomes, secretion of all of these byproducts. Sometimes we get formations of these, the book refers to them as nets, neutrophil extracellular traps. Basically, the neutrophils die. They explode all of their internal materials that form these like really thick nets to capture pathogens. It's actually part of the immune response. It's pretty dang cool. The neutrophils are going to secrete more chemokines and more cytokines, which are going to recruit now macrophages to that site of infection. And we're talking here about cytokines just as attractants. The other part of this is they are activators as well. They will enhance the activity of these cells. So there are some cytokines that when a cell comes into contact with, become voraciously hungry. They all of a sudden want to phagocytize every single thing with a PAMP. All right? And some cytokines will dampen or will stop this response, and that occurs later on. So the cytokines you can kind of think of, they're regulators of the immune system. The cells are already present, but the role of those cytokines is to regulate and to modulate how is this response going to occur. All right, I think I'll stop there for today. So please, a couple of things.